Good morning, folks. Welcome to day 2 of our Emotion Symposium. I hope everybody had a good night last night, fun, and after Chiara's lecture, good sleep. We've got a great lineup today. We're going to hear from Nolan Williams in a minute, and then Molly Crockett after that. And in between those two, we've got the breakout groups. So take a look at your programs, and you can select who you'd like to meet with, or actually rotate around to different rooms if you'd like to talk with different members of the faculty. But again, remember, the idea here is for all of you to let your hair down a bit and to get to know the faculty. And as Richie would say, drop your threshold for asking questions so that we can have a rich interaction. And just to remember, remind everybody, the lectures are about an hour, and then we've got a great 45 minutes for discussion. So we're looking forward to that. So it's my great pleasure to introduce Nolan Williams to you today. Nolan comes from Stanford, but didn't come easily last night from Stanford. And he just was telling me about the rough trip that he had, sort of the trip from hell, basically, but ended up finally ending up in Chicago unexpectedly, and then taking a rental car last night and getting in about 1 o'clock. So I've tried to infuse him with caffeine, but he didn't want much. So we'll see how it goes. All kidding aside, it's terrific to have Nolan with us, and he has really been a leader in the field of TMS for the treatment of refractory and treatment-resistant depression. Some of his more recent work not only is intellectually really quite interesting, but has very direct and important clinical implications. And as I'm sure he'll show us some of the data that he's derived from some of the paradigms that he's worked out at Stanford that relate to the use of theta burst stimulation given very rapidly over a short period of time with remarkable success in treatment responses. And we were actually fortunate in the American Journal of Psychiatry to be able to publish a couple of Nolan's papers, including a randomized clinical trial, which he'll talk to you about, both of which have received a tremendous amount of attention. So Nolan runs the imaging laboratory related to TMS and neuromodulation at Stanford. He's a recent associate professor, and comes from going to undergraduate school at the University of Charleston, and then the Medical University of South Carolina for his MD degree, and also there did a number of post-MD training experiences, which are impressive: internal medicine, a neuropsychiatry residency, a fellowship with Mark George, who was one of our earlier speakers here many, many years ago, a pioneer in using TMS for depression and in psychiatry. And then Stanford was fortunate enough to attract Nolan out there, where he did some additional research-related work, and then joined the faculty and moved up through the ranks, very rapidly. He's already been recognized with a number of awards. Most recently, he's received the Claremont Award from the Behavior Brain Research Foundation, and also a variety of other recognition from other awards, and also has numerous grants as well. So it's a pleasure to have Nolan with us. We're excited about hearing about his work. It also builds on the work that Rebecca was talking about yesterday from the standpoint of rapid-acting antidepressant treatments and how we can best leverage and put together different types of technologies. So let's give Nolan a warm round of applause.
Sir, appreciate it. Yeah. Great to be here. Like I said, like Ned said a second ago, I took a pretty serpentine route to get here. It wasn't a direct route. I had to talk the plane, the pilot of the plane, to letting me get off the plane in Salt Lake and not getting forced to Denver, where I'd have been stuck for 24 hours. So I am here because a pilot let me get off the plane. So I thank him. What's that? Yeah, I told him I'm a physician. I don't know. This thing's on, could you stop it? It's on some sort of timer. Yeah, there you go. Yeah, so I told him I needed to get here, and he let me off and let one other person. All right, so I'm going to talk to you guys about a technology that we called Stanford Accelerated Intelligent Neuromodulation Therapy. We've been working on this for about a decade. Now the clicker, do I need to turn it on or something? There we go. All right, so here are my disclosures. I do advise a company that's commercializing this approach now, but all the data that I'm going to show you, we developed pre-commercialization, pre-company. All right, so I'm going to give you a background on neuromodulation for depression, and you can kind of think about this across the board for neuropsychiatric illness, talk about something called theta burst stimulation, then talk about our rapid acting method. So for the first kind of grounding statement I'll make is, there are lots of ways to think about psychopathology. You all know that. You've been studying that. You probably heard different talks thinking about these different ways. But the way that we're going to think about it for this talk is to think about it as a condition of aberrant distributed neural networks. And it's really this idea, from my perspective, that we're entering into a kind of a third era in psychiatry and thinking about mental illness, with psychiatry 1.0 being kind of Freud and thinking about it from the, these problems from the standpoint of content, psychiatry 2.0, thinking about it from the standpoint of the synapse and neurotransmitters. And, I think psychiatry 3.0, we're entering into this phase where we're really thinking about these problems from a network level. And the great thing is it looks like, psychotherapy and meds actually affect these networks. So it's not like, we're giving away these two previous eras, but it's kind of enriched by that understanding, as well as this understanding that you can stimulate within focal brain regions. And if you think about the problem as a problem of distributed neural networks, the solution is probably a neural network solution, if that makes sense. And so I've studied depression as a kind of the primary condition for my career. I have some work in OCD and Tourette syndrome and Parkinson's related, neuropsychiatric symptoms, but depression's been the primary work for me. And there's a reason for that, right? It's the most disabling condition worldwide. You all probably heard that as well, right? Or have studied that, right? This is a highly disabling condition. You likely know somebody that's been depressed and had major depression, as you all know, and the drugs that we use to treat it aren't that great. They're not that fast. They don't seem to really consistently affect suicidality. They're not really geared towards high acuity settings. And that's where we've really been focused, and as I said a second ago, you can think about these problems as problems of distributed neural networks. We've known for some time. And I think Alan Schatzberg is a senior author on this paper. We've known for some time that, if you look at a depressed brain or a schizophrenic brain and you compare it to a normal healthy control, you see differences in brain network connectivity. And this is just an early example of that, where if you look at the subgenual to default mode network connectivity, the amount of connectivity is correlated between those two regions, those two networks is correlated with the amount of treatment resistance. And so as psychiatrists, we think about putting folks through these algorithms where we say with, STAR-D being a great example of that was a kind of a sequenced open label med trial. We put folks through these standard drug trials. We increase the kind of risk benefit ratio where there's more risk as you kind of change drugs, but the potential for adding benefit. And at some point they come to me. And so I don't really see patients until they've pretty much failed most of the standard classes of drugs. We haven't really focused on folks earlier on than that, and really are kind of zoomed in on those folks that are kind of at the end stage. And that's really the population I'll be talking to you about today. You all know this, but for medical students and psychology students that are just getting into this, people are very surprised at this figure. And I borrow this figure from a friend of mine, David Carrion, who extracted this bar graph out of this paper, which is essentially, if you look at moderate depression disability scores, it's as disabling as having an acute heart attack. Severe depressions is disabling as dying of cancer without treatment. And I had a physician one time say to me, I'd rather have my leg amputated than keep having depression. And he was serious. Or I'd rather have cancer because at least the family will circle around me and support me and understand what it is. But with depression, they have the same level of disability, but not the same level of understanding. And the hope that I think we all share, right, is the ability to get to a new level of understanding of these problems where we can see the problem in the brain and be able to show that to not only the patient, but the families. And so, again, I've been zoomed in on a very specific clinical scenario. And this clinical scenario is psychiatric emergencies. So emergencies where patients are admitted to the hospital and they need a treatment. So if you think about it, in the rest of medicine, if I fall down right now, I'm starting to have chest pain in here, you all will do a certain amount of things. Maybe somebody has an aspirin or something like that or whatever, and you'll do some things, but you'll then get me over to the emergency room. And you get me over to the emergency room because there are what? More tests and more treatments, ER doctors, right? And if I am truly having cardiac chest pain, I'll then get admitted to the ICU or to the cath lab, in which case there are even more tests and treatments and sophisticated doctors that are doing those tests and treatments, right? In psychiatry, it's the only place in medicine where as you escalate acuity, you actually lose treatment options. Think about that for a second. There are less treatment options as a generality on an inpatient psych unit when you're thinking about biological treatments than there are walking around as an outpatient, right? And there are no tests, right? We don't have tests. We don't have biological tests to discriminate, between different diagnoses and that sort of thing. And so what it really is kind of a safety procedure. But where the opportunity of that is that there is a kind of an infrastructure to build something, right? But what you have to build is you have to build something that's going to work within the context of the structure of how mental health treatment works, right? So it needs to be less than 7 days. And you need to be able to, as I've learned through sitting with various parties, reduce cost, right? And if it's a new expensive treatment, you can't reduce cost, nobody's going to pay for it. So you actually have to cut length of stay. And that's the trick, right? So you have to get a treatment in place where you can actually get people out earlier and in better shape. And this is an important one because the number one risk factor for suicide attempts and completions are previous suicide attempts and completion. We know that. That's not really a modifiable risk factor. The second one is inpatient admission, right? So going into the hospital is actually a risk factor, one way of thinking about it, for having a suicide attempt after. And actually the suicide lifetime peaks right after you leave the hospital. And it makes sense, right? If you go into the hospital and then you're told, you're locked in the hospital, and then you're told, yeah, there's no more medicines other than these drugs you've already been taking, right? And actually, if you wouldn't have done this, you could have gotten TMS and esketamine and all these things, right? So it's a really tricky situation. And for a lot of people are sitting in hospital beds like this out in ERs, right? So folks spend, sometimes a week waiting on this inpatient bed, in which case then they're told they don't have any new treatments, right? And they're very exposed, they're out in the hall, everybody sees them there. So we wanted to think about this in a different way, right? Can we provide an engineering solution for this problem? Can we engineer a treatment in the same way that cardiology is engineered treatments, in the same way that neurology is engineered treatments? Can we engineer treatments to solve this problem? And I'd say that we're about 1940, 1950s cardiology. That's really where we're at right now, just to kind of orient folks. Much good, right? Because we know what cardiology looks like today, but there are a lot of steps to take. And the first thing that they did was they made a transcutaneous pacemaker, right? They didn't make the implantable pacemaker first. They made this transcutaneous pacemaker that you can pace the heart exogenously. So they had a big box sitting outside outside the patient, and they had pacer pads on the chest. And they did that for about 10 years. And then they figured out how to do the implanted pacemaker. So I'm going to talk to you about a device that's kind of like a transcutaneous pacemaker and what we call a repetitive transcranial magnetic stimulation device. And so, the brain actually can receive information outside of your sensory organs and outside of its own internal processes. But it can only do so if you kind of interact with it in a way that is native to how it works, right? So you have to kind of drive it with an exogenous in this case, stimulation approach that has the same Morse code book that that we have, right? And so there are kind of steps to take for something like that, right? So anybody know how anybody study the telegraph and history? Anybody remember that? Somebody has. You do. Good. So what did they do first, right? They got Bob and Susie. Bob is in building 1 and Susie's in building 2, and there's a wire, and they click on the telegraph and they get it on the other side, right? So they get a signal that they made it there, right? So that's the first step. And so once they could click and get information, then they said, if you can click and get information, maybe we can make a code book where if I click 3 times, that's an A, and click 5 times, that's a B or whatever, right? And so Morse code was born out of this idea that you're sending a signal from one place to another. So TMS is really that, right? It's really just figuring out what the brain's Morse code is and then sending signals into the brain. So in this case, you're on one end of the can and somebody else's brain is on the other end of the can, right? Does it make sense? Right? And so there are going to be codes that the brain doesn't understand. That's good. That's like the sanity check. Because if you just put a bunch of information, the brain did stuff every time, there's no actual code book, right? So you need to know the language, right, in order for it to work. And so both parties have to speak English, or both parties have to speak Mandarin, or whatever it is, right? And so, the problem for us has been, we don't know what the code is, right? We don't know what the code book is. So you have to discover it. And the way you discover it is doing parametric experiments, right? And if you do parametric experiments where you vary the frequencies, and then you're able to use a readout that gives you information on what you've done to the brain, then you can actually get a sense of what is turning the brain up and turning the brain down. There's nothing else that we can do at this point still, right? We can't send complex information. We can't say sentences. Just simply say, turn up, turn down, and then we know sequences that don't do anything. Does that make sense? And so the old way of doing it was this kind of, 1 hertz, so 1 cycle per second, 2 cycles per second, 3 cycles per second, about 5 cycles per second. You flip from it being inhibitory or depotentiating or telling the brain to shut down to excitatory or potentiating or telling the brain to turn up, right? And that was the second discovery with TMS. The first one, that ping, 1985, the turn up, turn down, 1993. And then around that same time, Mark George, who Nev was talking about earlier, was at NDS, NIMH, and figured out, if you can turn the brain up and down, you can send the signal, and that signal means you're turning electrical activity on. And, he was in Bob Post's lab, and Bob was finding that there was hypometabolism, hypoactivity in the prefrontal cortex for depression. Gosh, maybe we can send this Morse code to say, turn on, turn on, turn on, and then that system will turn on. And that was the kind of the breakthrough aha moment. Everything I'm going to talk to you about after this is just an extrapolation of that, right? It's just an extrapolation of that viewpoint that we can send a signal into the brain and tell the brain to turn on in very specific brain regions. It's a very powerful idea, right? Because it's one that is very scalable, right? Much more scalable than anything else that we have in psychiatry. And so, that was kind of the aha moment. And then there was an evolution of things over the next 10 years where they kind of tweaked, subtly tweaked the dose and this and that. And they got to a point where they got this FDA protocol. And so it's very slow and inefficient but it gets there, right? About 1/3 of people in open label trials will go in full remission. 50% of people are responders, but this is not a treatment for emergency psychiatry. Getting back to my original point, right? It's something that's going to take 2 months to work, but in those 2 months, you can get a certain percentage of people better. And it's just the nature of things, right? It's like Moore's law. Like you're going to have a very slow computer at first until you get a better computer, but the computer was born, right? And then a third innovation was this idea of intermittent theta burst. And the idea behind theta burst, and this wasn't our idea, this was the idea of John Rothwell and published in Neuron in 2005, is this idea that you can send not just like what we call parametrically derived parameter set into the brain, but actually a parameter set that's based off of mammalian hippocampal biology, right? So if I put an electrode into a mouse hippocampal slice and I measure, I can see that there's a certain rhythm, so it's theta, gamma oscillations, theta, burst being gamma, right? So the burst, or the 3 pulses at 50 hertz every 1/5 of a second, is what we call theta burst. And really that's just a hippocampal rhythm. But the cool thing and kind of the crazy thing, right, is that we're just extracting biological signal. We kind of know what it does. And then we're sending that biological signal back into the brain. So we're decoding, and in the mouse lab, what the brain's kind of language is. And then through a TMS coil, we're sending that language back into the brain. And in this case, telling the brain with continuous theta bursts to turn down and stay down, kind of deep potentiation, and with intermittent theta bursts to turn up and stay up, right? And like I said, you can measure that using motor physiology, using EMG electrodes, right? You can get a readout of how much you turned it up and turned it down by how vigorous the thumb moves. And it's very reproducible in the sense of we can measure that same phenomenon across individuals and essentially turn the system up. And so that's been a surrogate. Now, some would argue not a perfect surrogate, sure, but from our parameter set, it gives us a way of decoding this information. And in this case, making the stimulation way more efficient, right? We're able to take what almost took an hour to do with setup and takedown down to a few more than 3 minutes. And there was a paper published in the Lancet in 2018 that led to FDA clearance. And that paper demonstrated that if you gave 3 minutes of theta burst just once a day, every day, 6 weeks, 5 days a week for 6 weeks, then you can produce the same antidepressant effect as conventional rTMS. And that's important to understand, because then that's a way of kind of telling you equivalence. And so we know from motor physiology and from those, kind of clinical application studies that the pulse, what I try to coin, I've never heard anybody use this term but me, pulse potency, maybe I can get Rebecca to use it, is this idea that theta burst, pulse for pulse, is about 5x potent, right? And in that Lancet paper, they gave 18,000 pulses per course. And I'll talk about that in a second. And in this case, there's been kind of an evolution. I didn't say this earlier, but the original targeting approaches were ruler measurements. So taking a paper ruler, measuring standard spots in the brain, says Dr. Kayla knows others. This is not, you can either pick the same spot on the skull or the same spot on the brain, but you never are going to pick both, so you've got to choose. And so they for expediency's sake, they chose the same spot on the skull, but kind of producing a different spot on the brain in each person. This study, they evolved a little more and took a standard coordinate and said, okay, what on average is where this dorsolateral subgenual circuit I'll talk about with you in a second. And so, they were able to show this was equivalent. We did this what I'll call engineering exercise, where we went back to first principles and asked the question of, if you're going to build a rapid acting stimulation approach, how would you do it? There's a lot of clinical and translational neuroscience that had happened since 1993. As you can imagine. And so one of the big things that they found is that there's a sweet spot of cortical excitability where if you do 1,800 pulses at 90% resting motor threshold, you actually get the best effect. If you start, and this is kind of interesting because this is what the original theta burst FDA clearance is, and seemingly it works better in geriatric patients because their brain's further away from the skull. But if you turn it up, you actually lose some effect. So really it's the sweet spot of dose and intensity. And there was a paper published, less known paper published in Brain, demonstrating that if you use a submotor threshold intensity with once a day ITBS, you get a similar sort of clinical effect, or even a better kind of effect than you do with the 600 pulses delivered over 6 weeks. And this is over 2. And then and the second question is, once you kind of know the unit and the session construction, right? So what does the stimulation look like in the time where the person's sitting in the chair, which is what we just talked about, right? You're giving theta burst for 9 minutes, you're giving it a certain intensity. Then the question is, how do you repeat it, right? The old way is repeat it once a day, and it's like 24 hours and you get it again, 24 hours, you get it again. The weekend, it's 72 hours and you get it again. That's not really what the neuroscience would tell us to do, right? How many of you have used note cards in studying for tests? How many of you have written one note card out and looked at it over and over again and set it down? One person. Okay, maybe we'll talk after. For most people, how many of you have written out like 60 note cards and looked at each one for a minute, and then you get back to the first note card after about 60 minutes? Yeah, so everybody's raising their hand. How did you know to do that? It's intuitive, right? It's like totally intuitive. So you all intuit a space learning theory. Space learning theory tells you that you have to be exposed, and then you have to wait about an hour to an hour and a half, and then get exposed again. And that's the optimal interaction. And so if you stimulate with theta bursts in hippocampal slices, you can increase the size of dendritic spines and prime some spines. If you do repeat stimulation in shorter time intervals, you get in mass stimulation. So you don't really produce any interval increases in dendritic spine enlargement, which seems to be a great proxy for some antidepressant effects. Ketamine does this. And so if you wait about 60 to 90 minutes, you have some dendritic spine enlargement, some priming, and then the next stimulation, you get more dendritic spine enlargement. You can actually kind of get a interval increase at each stimulation, right? We've done this in humans in translational work, where if you wait 15 minutes and you look at cortical excitability, you don't get much. If you wait 30 minutes, you get some change. And then there's work that's in review right now, or if you do the 60 minutes, you get more cortical excitability change, and that maps onto connectivity changes. And so I'll kind of stop there. That part is a reorganization of stimulation in time, right? This idea that we're using the same unit, pulse, but we're using it in a different organization in time. It's a different Morse code. It's a different code book. And we think a more efficient one, and we'll talk about that in a second. And then the second one is dose, right? So how many times do you need to expose the brain to telling it to turn on and stay on? How many of you think the more you studied the subject, the more you learned? Everybody, right? This is the same idea. You're just your brain's exogenously learning. And so it's of no surprise that we probably have a dose effect with TMS. And what's interesting is we just kind of did a certain amount of stimulation because of safety concerns, stopped. And then some work had been done, semi-recently, suggesting, well, maybe we didn't give enough, enough dose. Maybe if we just keep going and people that were non-responders, we can pick up more responders, and you start to see really plateauing the effect out at 4 months, way more than we were conventionally giving people. And so it's this idea that we didn't really do what the pharmacologists do. We didn't really do a dose response curve with stimulation, right? And I'll talk about that a little bit more later. And the third principle is a reorganization in space, right? So if you have the right signal, you need to send it to the right receiver, right? You need to send it to a system that is the disease system. You need to send it to a system in which, when it receives it, it's going to do something, right? And so, stimulating the brain isn't just stimulating at one spot in the brain. You're just, again, stimulating in a distributed neural network. There's a question around, if you're going to do a study like what we're going to talk about in a second, kind of how would you do it? You can't really do it in normal healthy controls. That's not ethical. So you do it in the target that we know the most about, so the left dorsolateral prefrontal cortex. And this is oversimplified for the imagers in the room, but useful. If depression is a problem with dysregulation of the subgenual DLPFC interaction, and normal mood is the dorsolateral regulating the subgenual cingulate, all TMS is it's just for depression it's just really a re-regulation of the system. We're really just sending information to the system to kind of re-regulate itself. And we're really just trying, I'll talk about this later, but we're really just trying to flip the signal, right? Not having the cingulate be in charge of the DLPFC. It's like having the prisoner in charge of the warden, right? And really it's the other way around. The dorsolateral is regulating the cingulate. And so, if you're, like I said earlier, if you're putting the coil in the same skull spot in everybody, you're not putting it in the same brain spot in everybody. But if you did a brain imaging, you did an MRI scan before you stimulate, but you just don't use it, then you can actually ask some interesting questions. You can go and say, okay, well, everybody's got a spread of positions. If some people just happen to be lucky and the coil was right over their ideal spot, and some people it was very far away, what does that mean as far as clinical efficacy? And what's interesting, and this is I think one of the most replicated neuroimaging findings in psychiatry, has been replicated 7 times now. But if you look at linear proximity of the dorsolateral subgenual circuit, if you're right over top of it, you get an antidepressant effect. If you're further away, you don't get as much of an antidepressant effect, suggesting that proximity to that target or depolarization of those cortical neurons that are involved in that subgenual target seem to be important. We dealt with this by using mid-level machine learning, so something called hierarchical clustering. And so basically we use hierarchical clustering to cluster similarly behaving voxels with a correlation coefficient of 0.5 within a named brain region, within the region of interest. So if you're trying to take a whole brain region and break it down into subregions and utilize each subregion independently to try to figure out within the bigger brain region where the coil's supposed to go, you can have machine learning do this, make subregions for you, and then apply a decision algorithm that'll allow for you to select which of those subregions is optimal. And in this case, we really wanted the subregion that was most anti-correlated with this subgenual cingulate sadness region, because we wanted an area that would, when you stimulate it, would suppress the subgenual cingulate and quiet the subgenual cingulate. So an automated targeting pipeline outputs the target onto the DLPFC. And then that's kind of the engineering steps. So again, reorganization in space, where, reorganization in time, how, and then dose, how much. And it's really just finding the right spot and saying, turn on, stay on, remember to stay on, right? And in a region that's kind of turned off from depression. So if you remember, I told you to remember these numbers. So we're giving 10 sessions per day of 1,800 pulses per session. If you remember, that was kind of the ideal cortical excitability dose. And so we're giving the FDA cleared dose for 6 weeks in a day. So various people told me this was crazy and why would you do that? And you're going to cause seizures. We'll talk about that in a second. But this idea that you're going to send a very dramatic amount of stimulation into the brain, a very short period of time, for the goal of doing, what I'm sure Dr. Price talked about before, yesterday around, rapid acting treatment effects, right? And thinking about kind of, inspired by ketamine, right? This idea that we can treat people quickly. And so we gave 90,000 pulses, or 7.5 months worth of conventional TMS in 5 days. And what we were trying to do is do this dose response curve. We were trying to figure out, is there a toxicity here? Is there a plateau of the curve in the right spot? But you have to, again, deliver it to the right spot to know that. Here's the, again, reorganization in time, space, and dose. Thanks to Ned and the editors at American Journal, 2 out of 3 of these papers were published there. The first one was published in Brain. It's kind of less known because we had a less fancy name for it back then. And so, we treated 6 people. In some ways, this is some of the most dramatic data because these were folks who met for deep brain stimulation. So if you're going to do some kind of really aggressive stimulation approach, you have to do it in the most severe sick people first, right? So we treated people that had failed ECT, failed conventional TMS, failed, 30 meds in some cases, and ketamine and VNS and all sorts of stuff. So we found this very robust but transient effect, non-durable effect. Now we've got papers being reviewed right now where we've given 2 weeks or 180,000 pulses, and we can actually right shift the durability of the curve, right? And if you give even more, stimulation where you give 2 weeks and then daily doses, you can right shift it more. So there seems to be a real dose effect, and it looks a whole lot like an implanted cortical stimulator, which I had been exposed to in my research fellowship, which is this idea that, if you keep stimulating in Parkinson's with a deep brain stimulator, you can keep having symptomatic improvement, in perpetuity. You have to adjust it a little bit, but for the most part, that's the case. And so I call this kind of beta saint, this idea, which kind of inspired what we were doing, that I worked with Ziad Nahas and Mark George to help do a 5-year follow-up on these folks that had implantable cortical stimulators. And they had a similar sort of effect, albeit a lot slower because the stimulation approach was not optimized. It wasn't, optimized based off the parameters that I described earlier, and the targeting was structural. But now what we're trying to do is we're trying to actually, and we're pretty close to this, we've rebuilt the firmware for an implantable cortical stimulator that can, or in this case, IPG, that can stimulate with saint-like parameters out of the stimulator and do, we think, exactly electrically what the magnetic stimulation approach is doing through the TMS coil. And then we can use TMS to pre-select study participants to put into an implanted study, which is cool because there've been a lot of failed deep brain stimulation trials for depression. But I think part of that is that we're not selecting the right people. You can use perturbation selection through TMS and like a crossover study and find only active responders, sham non-responders, and then put them in a trial like this. And we're really just emulating the spinal cord stimulation field, that's exactly how they do that sort of thing. But that's a digression, but kind of exciting. So then this is our first AJP paper. Ellie Cole worked on this. So about half of folks had a 5 or greater med failures. Their MADRS scores were like in the mid-30s when they came in. So very sick folks. And a lot of these people were actually Alan Schatzberg's patients that we borrowed from him. This is the HAM6. So this is pretty high for the HAM6. And so you can see there's a daily improvement. So this is the dose that we give for normal TMS. And this is an important thing, right? People say, well, conventional TMS, I don't know. It was just a kind of a dosing phenomenon, right? Like if you look at this, that's good. It's not great, but you got an arrow pointing towards, the really good stuff. All you had to do is just kind of keep going, right? And that's what we did. So this is first course equivalent, second course equivalent, third course equivalent, fourth course equivalent, fifth course equivalent. And so about, what is that? 18 weeks worth of TMS, you start to get a lot of remitters, right? And a certain amount of durability. And what's cool is if you take people that failed normal TMS, of course they failed normal TMS, right? If you look at the day 0 to day 1 dosing, they didn't get much change, but then you start to get it, but it takes longer, right? And if you overlap those ECT failing folks, they'd be doing this, right? It really was day 4 before they started to get better. So there's a kind of a dose phenomenon. There's also a relapse trajectory that's based off of treatment resistance, right? So, the ECT failing folks, they kind of do this, and then TMS failing folks, they lose it faster than TMS naive people. Treatment, retreatment, we don't see any tachyphylaxis. We have studies now where we retreat people out to a year. We can maintain anybody that responds or remits to a year with, with a MADRS score on average under 10. This is the average spot on the brain. If you measure the distance between here and the bottom dots, it's about 5 centimeters. That's about the same distance as the distance from this dot to motor cortex, which is the way we used to measure this. So really dramatically different positions. This is the randomized controlled trial that Ned was talking about earlier. So moderate to severe severity, moderate to severe treatment resistance, whole host of kind of failed things. And, we didn't want folks that had a history of RTMS just because of the blind. They'd be unblinded immediately. And then a history of ECT non-responders, not because we didn't think we could treat those people, but because they relapse so fast. So consort diagram, 2 people excluded, right after enrollment before we started stimulating. One had whole brain radiation, didn't tell us, and then the other one decided they didn't want to be on blinded trial. One person was excluded after they weren't transparent with us about their diagnoses. About 9 years in the current episode. So these are really, this is not trivial treatment resistance. These folks have been depressed were depressed for about a decade, about mid 20 year depression load lifetime, and about half of the folks were on disability for depression. And again, kind of mid-30s MADRS scores. These are response rates at the bottom. Cohen's d effect size is 1.3 to 1.9. It's statistically significant, however you slice it. And what's, all my kind of friends that are depression aficionados were like, how did you do this? And so what's interesting about this is that, I found in that Lee brain paper, they split up folks by level of treatment resistance in that paper using the Maudsley staging method. And in the mild treatment resistance, the active and sham curves were overlapping, actually. So there was literally no effect from active TMS because there's a floor effect from the placebo effect, right? But as you get into moderate and severe treatment resistance, you lose all the sham effect. And in the severe folks, they had no real, placebo effect at all. Their MADRS scores were the same throughout the entire time. So we really picked people that were pretty treatment resistant because we knew that we wouldn't have much movement and we wanted that delta. And so if you look at this, you can see that most people cross this remission line at some point in the 4-week follow-up. Now, I got some heat on this one because people didn't like this, but the reason why we chose this is because we think that this is a perturbation way of determining your biology, right? And if you think about it from that standpoint, it makes a whole lot of sense, right? I'm perturbing the system. I can get it there 80% of the time in these folks, and I'm only really getting it there about 10% of the time in the sham folks. So probably most of these people have that inherent biology, because the blind guess rate was chance. And then if you can get there, what we know now and what we're working on now is you can keep people there by redosing them. So it really is a lot closer to this, right? Because if I can take these people who relapse and I can redose them, I can flatten all of this. And that's what we're doing now. And that's what we think we'll do with an implanted device, right? It's that same idea, but it gives you the ability to know that's the biology and this is the right target for this person. Again, same distribution through the brain. We did this in a pilot in suicide inpatients, and we have a grant now to do this, a multi-site grant to do this across universities. These were pretty severe folks. They had 1.5 previous suicide attempts prior to being admitted to the hospital, 2 previous hospitalizations prior to this hospital, and they were all hospitalized on this Stanford inpatient unit. This is a retrospective. So this wasn't a non-inferiority trial. I'm not claiming anything. I just want to give you kind of an orientation, right? So if you take the folks that we treated and then you match sex, age, severity, and treatment resistance, match them with the ECT service at Stanford, you can see much more rapid effects. This is the SSI score, so scale for suicidal ideation. 14 is you need to be in the hospital. 6 is the clinically relevant suicide ideation cutoff point. So by day 1, 2, you drop them below the clinically relevant suicidal ideation. And in the people that had a response, they maintained it for 24 weeks. And so we can hold people. So this is my and Carl Dijsselhoff's postdoc, Anish Mitra, and Anish is superstar. I was telling Dr. Kalin about him earlier. And so he's MD, PhD, WashU trained by Marcus Rakel, one of the discoverers of resting state connectivity. So I was very excited when he got interested in our SAINT data. And he's specifically interested in this idea that if you look at the time series of bold fluctuations in 2 network nodes, you can see they're basically the same, right? They're basic. And so normally we would just average the entire time series of all these network nodes and say, this is a network, right? And so he said, well, that's nice, but I wonder if there is some differences. And he spent his entire MD PhD asking this question. And so can anybody, maybe the precocious student at the back, but can anybody pick out subtle differences in this time series plot? Yeah. Well, just call it out. Call it out. What's that? Like here? Oh, that's a good one. But even more, how about the even more subtle one? What's that? Bingo. Bingo. Exactly right. And so if you do this thing called parabolic interpolation, because the TR for this is 2 seconds. So the time resolution on MRI is not like EEG, right? EEG, really good time, but not good spatial resolution. MRI is not good. Time resolution would be good, spatial resolution. But if you do this parabolic interpolation, you can interpolate a peak. So you can see, in this kind of cartoon example, if you just simply ask what the peak was based off the data, you'd say it was at the 0 point. But if you kind of interpolate the peak, what it would be with mathematics, you can basically see that it's going to be temporally in front of that. So if you take these two bold time series and you interpolate the peak, blue is always in front of orange or red. I'm kind of colorblind. I think that's orange. And so you can do this for each voxel. And when you do that, what comes out is that if you seed the dorsolateral, comes out that you get, or the cingulate, and I'll show that in a second, then you get this temporal offset in the other, right? And so in normal healthy controls, the dorsolateral precedes the cingulate, kind of inferring causality, right? So if something comes before something else, it's likely that it's causing that thing. Now, we can't prove that with this experiment. You can do it with other experiments. So, we seeded the dorsolateral. We asked this kind of question baseline, 1 week contrast, and you get this cingulate temporal offset, right? So in people that received active SAINT, you see that there's a dorsolateral cingulate temporal difference. If you do the same thing with a cingulate, then you get dorsolateral anterior insula. We repeated that with our open label data from the 2020 data set. And then we looked at the correlation with the clinical scores. And interestingly, the dorsolateral doesn't have any correlation at all, but the cingulate does. The cingulate has a correlation of this temporal shift with the MADRS score. And then we took our depression pre-treatment data, and then we compared it to our normal healthy control data set and the WSoE normal healthy control data set collected by Randy Buckner. And we contrasted A to B, which is D, and A to C, which is E. And you can see that in both of those, that if your seed is a cingulate, then the cingulate is temporally in front of the dorsolateral and the anterior insula. And then if you take the SAINT high responders, the 90% responders, and you contrast them with the folks that had no response, you see the same thing. You see the same areas highlighted, right? That the anterior insula and the DLPFC are temporally offset from the cingulate. And what's kind of cool is there's a correlation of the amount of this lag and the MADRS score on the pre-treatment, suggesting that, the worse the patient is clinically, the more. Now, it's not super strong, which you would imagine if it was super, it'd be probably not true if it was totally correlated, because these things are not, not probably that reliable. But this idea that you get some correlation between the lag and the MADRS suggests that this is kind of also a biomarker, not just the presence of a subtype of depression that's SAINT responsive, which is cool, because it, on the post scan, as I said earlier, you get a flip of the signal, but that it's correlated with disease severity. Okay, lots of folks that are collaborators and students and mentors and past folks, and this is the lab. Actually, finally, Stanford dropped the mask rules, so we can actually take a real lab photo soon. And this is all of our funding. Yeah, thanks, and happy to answer any questions.
Thank you, Nolan. So I want to invite Katie and Hadley up here to.
Great. Hey, how's it going? Good to see you.
Thanks so much for this talk.
Yeah, of course.
Bye. The gray button. Oh, okay. Thanks so much for the talk. The first question I kind of have for you is, you talked a little bit about like responders versus not responders,
Sure.
Sort of after initial like TMS kind of treatment. And have you thought about or have data on predicting like forming prediction models,
Yeah,
basically.
that's what that last bit was actually.
Okay.
Yeah, so we we're going to try to now prospectively predict, we kind of built this out, and now we're going to in the new data set that we're collecting for NIMH prospectively predict based off of the pre-treatment scan. If they have this, they look like, this, we would predict that they had they'd have a pretty, because it's the pre-scan, a pretty robust remission versus this group, which are the non-responders. These are depressed people. They're non-responders, and they look just like the healthy control data set, right? The non-responders, SAINT responders, look just like normal healthy controls from this temporal biomarker. And so we didn't see any clinical improvements really, and minimal clinical improvements in those guys, 10-20%, whatever our placebo rate is in the sham group, so.
And then what about predicting, like even prior to eliciting treatment, like who would be treatment resistant in the first place? Like
Yeah, that's what
demographic data, hospitalizations, like all of that in a like larger prediction model. I know you need a lot of data for that kind of machine learning, but I can see that you kind of do machine learning and engineering techniques in other ways.
Yeah, so there is an effort right now based off of psychometric and biometric data to predict the MADRS score, and we have a machine learning algorithm that can predict the MADRS score with pretty high accuracy for triggering retreatment, but you got to train it on the patient's data, right? You have to show it remission and not remission and all this stuff, right? So at some point, you'd be able to do that before it got, but right now you kind of have to show these machine learning models what the contrast is, and it has to kind of seemingly learn that, and then its accuracy goes up as it sees them. I mean, that's what's really nice about SAINT, right? Is that you have the ability to have this pretty dramatic contrast, and so as a kind of a translational neuroscience tool, it's cool because, in a big, and basically the entire population, I think ethically, unlike ECT and other things, ethically, you could do this in a treatment naive person. You could do it in the most treatment resistant person, right? And you'd be able to kind of get a sense of that. But that's how you'd have to train these, these machine learning models, right? Is you have to kind of give it that contrast right now. And so the goal eventually is to get to a point where you can do what you're saying and predict before any information, but to do that, you kind of have to do it at scale. And so that's why, as I said at the very beginning of the talk, like all this tech went out of my lab into a company, and Brandon Bensley and Ellie Cole, who are the postdocs and research track residents that were first authors on the AJP papers, are all working at the company now. And they're all working to do this, right? They've got kind of mind-blowing data on this kind of thing to be able to try to get the next step. And it gets to this point, this kind of bigger point of how do you get the sorts of things you're talking about at scale. You have to be able to do it like not just in your lab level, right? You got to think beyond that. And so it's like at this level of scale of all this information coming in, from a clinical enterprise, right? And being able to take all that imaging and biometric and psychometric data and being able to process it for the purposes of kind of triggering treatment and that sort of thing.
Yeah, thank you so much for that. I think that makes a lot of sense. And sort of thinking about like the larger scale of things, how do you see this kind of becoming utility in a more broad scale, like medical setting, is particularly for low resource settings, as I would assume this could be costly, and people that might not have insurance and other kind of coverage like that?
Yeah, so I just found out 3 days ago that that the SAINT NTAP application for Medicare went through, and what that does, which is interesting, is that it will triple the reimbursement for inpatient admission for Medicare patients, right? And so they'll, the average Medicare reimbursement for an inpatient is $10,000. And so it provides $20,000 more to do this in an inpatient setting. And what's cool about that is, it's that Medicare population, maybe on disability, maybe older folks, and it gives it kind of gives access to that group first, right? Even before private insurance. And the cool thing about Medicare is they cap an MRI charge at like $400. And if we can predict who's going to respond ahead of time, like it's done. Like anybody will, like they all want to pay for that, because if an MRI predicts that 2 or 3 out of 10 people aren't going to be remitters from this, it saves the money for those 3 to run. For the other 7, it pays for all the MRIs, right? So it's this kind of meta cost thing. And so, people worry so much about this MRI thing, but the MRI thing actually is going to be cost savings at the end of the day. And so then it's good, if you can extract that much information out of an MRI. And then the goal is, if you can cut length of stay down, you can cut costs down, and then it can be broadly applicable to people.
Great, thank you.
Yeah.
Thank you for your wonderful presentation, and thanks for being here today, considering all the trouble.
Yeah, no, I'm glad I'm here and I'm not in the Denver airport. I can tell you that with 100% certainty.
Yeah, us too. All right, so many of your studies employ samples as small as an N of 5 or 6.
Mhm.
So as scientists, we're well informed about the limitations of low-powered hypotheses, but we rarely consider the potential advantages of small N designs, which are sometimes unavoidable, as I'm
Yeah.
sure you know. Reading your work, I can't help but notice that you and your colleagues have access to a great detail on the life history of these patient participants, which could be highly informative. So with this in mind, what else can we learn from clinical trials with these small sample sizes?
Yeah, no, it's a great question. I mean, just to give people a context for this. So this was a grassroots effort, philanthropy funded. It's, I think, one of the few examples in medicine where essentially patients paid for their own treatment to be developed through my lab, right? I mean, I got money from NARSAD, which was 1/20th of the overall amount of money it took to pay for these trials, but the vast majority of this was a grassroots effort from patients' families, patients' friends. And so, to your point, right, at the end of the day, we think about these sorts of things, it's, people are hyper-focused, p-values and that sort of thing, but, it's really kind of cones the effect sizes, right? And how much of an effect you're having. And it, in some ways, is related to the kind of, signal to noise problem of with large trials, you end up having to enroll people that aren't well characterized, to your point, and folks who you don't have good deep histories on. All of these patients, we had medical record reviews, we had like big conference discussions, we discussed every single patient. We made sure that we were looking at a treatment-resistant depression patient. Multiple people looked at them before they came through, because I was, kind of acutely aware of this issue, right? And we knew, because of the funding limitations, that every single person had to count, and it's super expensive to kind of run these sorts of really heavy sort of stimulation approaches. And so we were very cautious to bring people in. I think that what that tells you is it's a subsample of the bigger sample of what we call depression. And my suspicion, the number of these non-responsive folks is quite low in the trials that we ran so far, but it's likely that if you do this in a broad scale, to your point, what you're going to see is a drift, right? We're going to have more of these folks in the sample. And I think that's actually okay, right? If we have the problem with kind of a blind clinical trial without the biology in it, is that the non-responders don't tell you anything. But in this case, to your point, right, the non-responders can tell us a whole lot, and we can kind of get a sense, because it's probably that we're dealing, as many of us think, Connor Listens talked a lot about this, of multiple depression, what we call biotypes, right? Or multiple depression kind of phenomenon that are occurring that are under that depression umbrella. Now, with clinical rollout and with everybody getting a scan and with everybody, getting, clinical scales and that sort of thing, through this commercial effort, through a lot of our, our NIH grants and that sort of thing, we're going to have 1,000 patients worth of data by 2025, 2026. So, this question of, of low sample sizes will kind of go away, but it is kind of a win in my head for this idea of, if you pick the right patients, you can do a whole lot. And, with, I think we submitted, there's another study that got run after the IP went out, but I think it was like 60 people got submitted to the FDA for a clearance, right? And then they went for it because they understand this idea of kind of separate, the degree of the separation of two groups and all that good stuff. So
Yeah, it sounds like to a degree, these early patient participants sort of like self-selected into the study.
oh, yeah. I mean, they self-selected to be screened, but we
Sure.
definitely screened out a lot of people. If you look at the consort diagram, like we had an online, it was bipolar patients and people with schizophrenia and all sorts of other stuff, but we had like a 10-to-1 conversion, right, of people who got screened out online that we would talk to. And then when we thought they had treatment resistant depression, it was a 2-to-1 conversion into the trial.
Yeah.
So it was pretty strict actually to get in.
Absolutely. That's only to say it almost sounds like a study that's almost partnership with participants.
Yep. No, that's right. Yep.
And I'm just curious about whether you got any pushback towards that, considering, there's lots of pressure scientifically to sort of be a blind observer.
Yeah, I guess I'm not following you.
That's okay. Just because there's often pressure, like in scientific trials, for researchers to be blind, to conditions or to not have any expectations about participant outcomes.
Oh yeah, we had all that. I mean, when I said earlier about it being funded by families and stuff, that's way after they participated in the trial. Yeah.
No. Yeah. I'm just interested in the merit of being partners with your participants.
Yeah, no, we're definitely partners. I mean, every single person, so we do the trial and we're very rigorous in the period of time when they were in the trial, as far as all the things that you're describing and you're worried about. But then, once people got into the trial, we made sure that they had some sort of long-term game plan and follow up and all that good stuff for, continuing to get treatment and all that good stuff, as far as that partnership, if that's what you're talking about. But the actual trial itself, we had to be obviously extremely careful and methodical
absolutely.
about blinding. All that. Yeah. So
Yeah. Thank you very much.
yeah, thanks.
So maybe we'll open it
up now and we can come back to you guys if you have more questions. So let me start, Nolan. And given the role of dorsolateral prefrontal cortex and subgenual ACC that is featured in this work, have you guys started looking at the specific behavioral markers and more granular symptoms that might be specifically affected and show rapid change? I mean, at a very coarse level, for example, do you see an initial reduction of negative affect, or do you see an elevation of positive affect? Do you see both? So I'm just really interested in, and also interested in, given the specific parts of the network that you're describing, if there are particular symptoms that you would hypothesize to be most dramatically affected as a consequence of this strategy.
Yeah, definitely. That's a great question. And so we've been funded by NIH to do, and this is quite the study. So to do daily scans on people getting sane. So we're going to have 10 scans for each patient, 2 baseline scans, one a couple of days before, and then one like right before they run, and then every day after for 5 days, and then what is that? So it's like 7, and then I think 3 more once a week or something like that out to a month. So we have a ton of, we're going to have 1,000 scans on 100 patients to characterize just this, with clinical scores every day. Because and this is unpublished, but we're, I think, going to submit it soon. Lassitude seems to be the thing that moves first, which is interesting because clinically I was seeing their SI go away first, but when my fellow looked at this, lassitude seems to be the first thing to go. And then some of the other symptoms follow. If you ask the question of, because we did Madrus's and Hamilton's, if you ask the question of which items contribute to the change score, in the Hamilton, every item contributes, in the Madrus, every item contributes to the change score. But in the Hamilton, only like 10 or 11 of the 17 items contribute to the change score. And there are some symptom scores that just don't change, like anxiety and stuff, but it pretty much maps onto another, like plugging AJP again, another AJP paper that Sean Siddiqui and Mike Fox wrote, where they had two different DLPFC targets, right? And they had this anxiosomatic target and this dysphoric target, right? So if you take our coordinates, you plug it in to their map, it's overlapping on the dysphoric target and the symptom changes are the same. And if you look at the anxiosomatic symptoms, they don't really change so much. But again, the problem is to your point, small sample sizes, it's hard to get a good sense of this until we have the data at scale. But I think what we're going to see is, to your question, Rishi, a temporal progression of symptom change that may be, kind of triggering us to, as another way of kind of triggering us to understand this person's going to get better or not, because they get a lassitude change first or not, but still a lot to do.
Let me follow up, Nolan. First of all, congratulations on the work. It's really exciting, and especially the lag correlations between different brain regions. And just to be clear, did you say both DLPFC and insula in relation to ACC?
Yes.
Can you describe that? So both are typically ahead of ACC in normals, but behind in
depression.
Okay.
So you can see it.
And what about DLPFC to insula?
I don't think I can ask if he did that.
Okay.
I mean, I think they were both kind of lagged at the same level, but I can ask him if he saw any temporal differences there. But yeah, that he, Anish is very excited about this idea that to stimulate anterior insula. It's probably your thoughts too, right? Use ultrasound to try to do that. But yeah, it's it's both. And in the bottom contrast here, you can see both. So when you see it, you can see it.
So the finding is really nice. I'm particularly interested in the non-responders.
Yeah.
And wondering what you know about, circuit alterations in relation to lag correlations in them. And also, what do you know about less depressed individuals? So not treatment refractory individuals, but the general population depressed individuals in relation to this timing of correlations. Is this found there too? Or is this?
Yeah, no, it's a great question. So he hasn't done that yet. Leanne Williams has that data, and I think he's going to do that soon. I think your question is, at some level is this a phenomenon of treatment resistance, or is this a phenomenon of severity? I can't remember actually. I asked him at some point, I don't know if he did it or not, but I'll ask him again to run the level of treatment resistance with the lag and see if there's a correlation there. I'll ask him that, because I forgot about that. I asked him to do it, and then I forgot to follow up. But he did get some correlation with the level of severity. But it would be interesting to kind of get that sense of things, right? Is this a treatment resistant phenomenon? Is this a acute severity phenomenon? Is it something that's more scalable out to the general population or not? And I kind of on that same line of thinking, right? We know in epilepsy that if people have mesial temporal sclerosis on their brain MRI, then you can give them drugs all day long. They have like a 2% rate of having an anti-epileptic effect from a drug. And so the epileptologist will just go straight to surgery in those cases, or neuropace or whatever. So you could make an argument in these sort of scenarios that, if this phenomenon present in individuals, and then for some reason they're not, say, drug responsive or something, then you could make a similar argument here, where you go straight to something like Saint and skip the drugs, and that's one of the big questions. I mean, it was, the data set is limited. We need to do this again. We need to do it in a bigger way. We need to do it in more samples. yeah, it's pretty cool.
Thanks.
Thanks.
Other questions?
Hi, so it's really amazing to see how close you seem to be to getting this to patients. Like
Yeah.
you just discovered it recently and now you're on, the Medicare news is amazing. As
Yeah.
my understanding is if you get Medicare, then private insurers will often follow suit. So
That's
that's
right.
huge.
Yeah,
So it
thanks.
sounds like, I'm just curious about your estimate on the time frame here. So it sounds like maybe for inpatient settings, it's going to be rolled out in the near
2024.
future.
a
And
year.
then what would the next step be? Outpatient providers who have TMS equipment and access to fMRI or?
Yeah, I'll tell you, I've learned so much about billing and coding over the more than I wanted to learn, but that's okay. It's definitely a good skill set to understand. And so there's this phenomenon called a medically unnecessary edit. And the medically unnecessary edit is this coding structure within Medicare that prevents you from editing the code unless like President Biden goes down to the office himself and says, you need to change this or whatever. That probably wouldn't even work. But so it says you can only give one and only one a day of TMS for the outpatient codes. And so you have to come up with a whole new coding structure for this. And the APA has to be on board with that coding structure. And so outpatient's actually going to take a while, multiple years, but inpatient, there's never been TMS. And so you set the rules of the code. So then you can say stimulate 10 times a day and all that stuff. And there's no previous code with a medically unnecessary edit in place. And so then it's much easier. And there's a structure for Medicare to pay for inpatient interventions much quicker. And so it looks like what's going to end up happening is that inpatient is going to get covered very soon in a year. And then that gets scaled out. And then there's like a ton of work that needs to get done. And to get a new code, you have to have 700 patients worth of data, right? As we were talking about earlier, right? And so you have to get, like 5 independent publications, all this stuff, and then you get a new code. And so then the outpatient will take, 3, 4, 5 years. And so I think that's kind of the timeline. But, our goal was to make an inpatient move. So, we're kind of happy that the inpatient thing is going to work out sooner. And then we kind of go from there, but it'll give some time too. Cause the problem, as right, is that getting a multi-band, multi-channel MR head coil in a clinical space, some of these hospitals have them. They're using them as like a, whatever, like put their Cheetos in or or whatever, like they never touch this thing. It came with their MRI scanner and they didn't have any use for it, right? And this is going to be the first, this is the first MRI clearance by the FDA for anything in psychiatry, the first time resting state's really been cleared. So it's like one of these things where, getting that clinically rolled out in an outpatient setting, it's actually going to take a little bit of time anyways, because of these structural limitations of how the scanner thing goes, and really getting collaborations with radiologists, right? Because, that's also something, psychiatrists will interact with radiologists, and those of us who do imaging will interact with them more. But, to have essentially a pretty regular involvement of radiology in a psychiatric care, that's new, right? So that's going to take some time too. So I was able to talk Stanford Radiology into giving me a courtesy appointment to help with this, to be like an ambassador of these things. So we'll see how that goes.
Thank you so much for the wonderful talk and risking your relationship with the TSA to be here.
There you go. Yeah. Yeah. It was really interesting. Actually, they had to radio me into the control tower and get clearance to let me loose. So it never feels weirder than being told you're stuck on a plane and you're not allowed to get off. So
I bet.
yeah.
So please correct me if I'm wrong here, but it seems like TMS largely operates through Hebbian forms of plasticity.
Yes. Yep.
So my question is, I'm curious if there are any interventions which operate through non-Hebbian forms of plasticity, which could be used to, say, supplement TMS.
Yeah, no, it's a great question. And these are, we're at the very, very beginning, and I hope that we're not doing SAINT in 30 or 40 years. I hope we're not, because if we are, then things haven't gone the right direction, right? The goal is platform technology that can kind of either get supplemented, augmented, changed, right? And so, we know that you can do, for instance, priming stimulation and make the brain more receptive to theta bursts, right? So you can do TDCS and you actually make it more receptive to theta burst or normal TMS and have a greater propensity of having an anti-nociceptive response or changing cortical excitability. We know there are things like coordinated reset, which the goal is just kind of to trigger the brain to kind of reset, right? So it's kind of an acute stimulation approach that causes kind of what they think is kind of a reboot of the system or whatever. And Peter Tass at Stanford has been working on that in various forms. And so, the problem ends up being, how do you decode the system to understand it, to be able to figure out what you're asking, right? Because at some level, we're kind of in a pitch black room and we're reaching around for things. Oh, this works. Okay, we're going to go with this for a while or whatever. And that's why I think, I've been very focused on making these implantable cortical stimulators that have recording capacities. So then you could have machine learning algorithms just cycle through. And if you've got a readout, say we were able to get an electrophysiologic version of this, to get kind of a sense of it, then, we could start asking those questions in an organized way, but it takes a microscope to ask the question, right? And I think that's part of the problem. And so we've been in this chicken egg sort of thing where we can't really get a handle on the neurobiology. And so a lot of people are just like, let's just I'm involved in some of this, stick like 10 wires in the brain with 100 contacts and just record, right? Helen Mayberg's doing this. The folks at Baylor are doing it. We're going to do it in OCD patients. And that's one way to kind of deal with it. We've taken a much gentler approach of trying to do this, but it's the same goal. It's really perturbation mapping, and trying to get some readout that's reliable, and then trying to take that readout and then do a bunch of different parameter evaluations to see if you can get that readout. And that's what I'm saying. It's the chicken and the egg. So we've said, okay, we got something that's functional. We can move some of these people. We can take these folks that have the biomarker, we can move them. We can understand that. And then if you've got a biomarker that seems to kind of select and move, now you've got an opportunity to try other parameter sets and see if you can move it too, right? So you can get more information, something that's a reset, like a coordinated reset. That's the non-Hebbian sort of phenomenon. But to me, it's like getting to that point and then being able to use that to then kind of modify it. So it's a hard game. I've taken a shot at how to play it. I don't know what the right answer is.
May I ask a follow-up?
Sure.
So we learned yesterday that sleep may promote non-Hebbian synaptic reorganization. So I'm curious if something as simple as sleep following intervention may be able to explain some of the variance in responsivity.
Definitely. Yeah. And, there's a lot of work with, with suicidality in sleep. And we know folks that have insomnia don't do as well from a suicide standpoint. So, absolutely. And we made sure, that folks were getting good sleep, that they had good sleep, if they had insomnia, they were getting treated for insomnia before they started the trial and all that stuff, because we wanted to make sure, because for Hebbian plasticity, you too need to have a good sleep consolidation, right? So it's kind of both actually. And so it's hard because we're sensitive to that for these trials. It's hard for me to tease that out. I think that there's a world where you could do a trial where you looked at that to try to get a sense of it. But, I don't have that because we were sensitive to that. There's no real way for me to kind of sort that out. Anybody who wants to come and do a postdoc with us, let me know. And that's definitely, yeah, that'd be great. Yeah, we should talk. Cool. All right.
Thank you.
Yeah, no problem.
Thanks for a great talk. I'm just thinking about sort of synergizing your talk with Dr. Price's work from yesterday. And I was curious if your patients were simultaneously getting any other kinds of treatments and whether or not they were, whether you would expect interactions between this kind of intervention and a more psychopharmacological or psychotherapeutic approach in treatment-resistant patients.
Yeah, that's a great question. So we didn't, the problem with medications in patients and all of this sort of thing is, patients will tell you all day long, oh, this med I'm on, it's not doing anything for me. And as soon as you take the patient off the med, they're like peri-hospitalization, right? Like, it's just enough to kind of keep them out of the hospital or something. And so, in a severity group like this, we didn't want to do kind of a monotherapy sort of move where we washed people out and then did stimulation, because we were looking for this kind of end stage, very severe cohort, which is a little bit different than normal TMS trials where they were taking folks, who had been, were kind of getting switched on to a new med. So they waited to do the switch and then they came in the trial and they were kind of more on the 1, 2, 3 med failure group sort of thing. And so these are folks who have stable meds for 8 weeks prior. And they, and if they were getting psychotherapy, they were getting psychotherapy, ongoing. And so their baseline was a baseline of everything that they were doing right before they went in. And so it is that one, that issue of, even though we don't think the med independently was doing anything, there's an open question about, is there synergy that occurs between stimulation and med, and these are open questions, right? We don't, and that's also kind of like a super early field, right? There's some data with D-cycloserine on this, whatever, but can we optimize by combining stimulation with meds? Can we optimize by combining stimulation with psychotherapy? We've got some work that we're getting out now to show that we can change, trait hypnotizability with TMS, with this idea that we can potentially get people more prone to have a therapeutic effect from hypnosis. So it's these open questions of these interactions between treatments, and that's super early. Like I always tell students, like, you have a job for a long time if you want to study this stuff, like nobody knows most of the answers, it's like super early days on all this. And, a lot of studies need to get done, as far as it goes. And I think it's, for us, we've just been trying to build tools,
Yeah, I had a question that's sort of along the same lines as some other people have been asking about, can we identify sort of the behavioral correlates of whatever this biotype is? And so I was wondering if in these early stages, if you're collecting any qualitative data, and if there are potentially any insights in the patients have about their experience that maybe aren't captured exactly with the kinds of questionnaires that we have that aren't designed to probe this circuit.
Yeah, no, that's great. That's a great question. So we have been in these current, COVID was very nice that was not nice, obviously, but it was very nice for reorganizing your lab when things went down, because we were able to spend some time doing some of the stuff that we couldn't do before around trying to get new procedures in place. One of the things that we put in place during that period is doing video capture on everybody for every assessment, and trying to get, for the purposes of what you're saying, right? Yeah, the tone, the affect, the facial expressions, all that, in addition to the additional content that they give you, because as you know, whether that's structured interview, MADRS, or Hamilton, or whatever it is, you're going to ask questions, but then people are going to come and say other things, right? So we can have all of that content kind of put down on paper and extract some of the subjective information. So we're trying to do that now, and trying to see if some aspect of voice tone, facial expressions, additional content that's outside of the structured scales gives us info. What I can tell you is we had an extremely rigorous clinical screening process, and we still had people that had, this, right? So like, we don't know how to pick these guys out clinically if we could, and when we wouldn't have had this paper, right? And so it's one of these things where, you almost need to have the biomarker. So you have two groups, and then you can do exactly what you're saying and go back and look and say, okay, now that we have these two groups and we have all this extra data, is there something consistently in this group? Is there something consistently in this group? And that's why I always like to use this term of perturbation mapping, right? We're perturbing the network. We've got this finding, if we didn't have the Saint data, what would have been interesting is we'd have had people, this would have been a terrible biomarker to find, right? Because you have people that that have it, you have people that don't have it, and then you have the healthy controls. And so, be like, this isn't useful, right? Because you're like, like only some of the people have it, and some people look just like a normal healthy control. Well, that's useless, and throw it away, right? So you need the perturbation mapping to say, okay, only in those people can you stimulate and kind of align that information. And so that's why I think neurostimulation is a very powerful tool.
So, hey, Nolan, around those lines, you well know that biomarkers have suffered from reliability and also replicability.
Yep.
So what can you say about the reliability of the lag correlations that you're seeing within an individual? I know you're going to do the study where you're going to get 10 scans or whatever it is and all that, which will help. But what can you say now about that?
So replicability, I can kind of address that first. So we did this in the second study, and then we looked at the first study data. So we were able to see it across two cohorts. I think we need to have multiple data points, in each individual to really source out the reliability question. And we hopefully will know that in 2 years, I think,
Good. Other questions?
Yeah, thank you for explaining all of this so clearly and also relating it to the policy implications. I have to say, I don't think I've ever heard someone start a sentence with the cool thing about Medicare is.
I wouldn't have said it 2 years ago.
Right. I actually wanted to ask a question going back to like the basic biology. You mentioned that theta bursts are a feature of mammalian cortex. Is that specific to mammalian cortex? Is that something you see like
mammalian
emerge
hippocampus.
Okay.
Yeah.
It's not something like emerges in cell culture or that you see in other networks.
Yeah, I mean, this is reasonably specific to kind of recordings within hippocampi and mice all the way up, right? And so I'm not aware of those sort of theta gamma oscillations happening, in cell culture, but I could go back and take a look at that. But it's, from my understanding, it's really, you have to have some kind of connectivity of these circuits together to really get that emergent property.
Okay. Yeah. Thank you.
Yeah, I was curious about, considering you were talking about lags, I was curious about use of EEG, which might help in scale. And, because the temporal dynamics are much more efficiently captured there.
Yep.
And so if it would be worth, because instead of 10 scans per patient, not instead, in addition to 10 scans per patient and so on, but if you have EEG data, then you could probably compare and see if, there is something which very reliably tracks the findings you have. What do you think of that?
Yeah, no, it's a great question. So we started out this NIH trial, and it wasn't part of the scope of the trial, but we were going to add in EEG just because of this question. And then like my lab had an intervention with me about this, and and told me that the patients, you get somebody, they're going to wake up every morning and get scanned. I mean, it's a lot, right? And so it's one of these things where it's just not really, trying to do all that in the same patient population is unreasonable. We have a different study we're doing some EEG on, but what we're really, it's a great question to your point. What we're really interested in now, and Anish is looking at our OCD Saint data, and we've got this study that with this foundation called the Foundation for OCD Research. Rob Malinka, who's at Stanford, running this, so I was able to talk him into like merging two studies together where we took people, they have to go through our Saint protocol with OCD, get that, and then the folks that have some sort of response will then go on to get surgery and get stereo EEG electrodes placed. That was what I was talking about earlier. It's like 10 wires, 100 contacts. And so, Anish is right now analyzing the OCD data. So if we can see a temporal lag, and we could see it on EEG and MRI, and then those same people are going to go get a deep kind of phenotyping of electrophysiology to try to get a handle on the directionality and see if the kind of signaling directionality in direct brain recordings validates what we're seeing in neuroimaging. And so TBD, that's probably a 2027 paper. And I'm hoping that, I'm hoping something's there for the reasons that you're pointing out, right? Because it'll help to kind of validate this as a real phenomenon if we had an electrophysiologic correlate. He's seen these electrophysiologic correlates in mouse studies in normal healthy epilepsy patients just have epilepsy and nothing else that correlate with the imaging, and they have stereo EEG electrodes. And so there is some validation there, but to get it in the disease model would be great.
Ned, I think there's a question over Zoom, potentially. I think there's a question over Zoom.
Yeah, I wanted to ask just one thing, and then we can go to the Zoom. Nolan, the circuits that you're talking about and some of the parameters that you featured, like the lag parameter, are presumably normally distributed, and at least they're continuous. And that's being mapped onto a categorical system for classifying psychopathology. And I wonder to what extent some of the underlying neural systems that you're describing really can be thought about more continuously, and that there are individual differences in these parameters that may be associated with patterns of emotional reactivity, vulnerability to depression among people who may not have as extreme a set of parameters. And can you envision a time where these kinds of neurostimulation methods might be used in more subclinical individuals, perhaps in a preventative strategy, to prevent the subsequent occurrence of more serious psychopathology, to stabilize these networks, if you will?
Yeah, that's a great question. So I'll answer the first, I think, kind of two questions. The first question first. So it's about 100 people, right? Between these two groups, there are only 2 people that had the lag, which is kind of interesting. And it's like a directionality. So it's a flipping, right?
Reverse lag.
No, 2 people who had a cingulate first in the normal healthy controls. Yeah. My suspicion would be those people probably were not answering the clinical score correctly, but clinical scale correctly. But I could be wrong about that. And those folks, this is a kind of a phenomenon happening in some healthy controls, but it's 2%. So it was really low, and every other person had the DLPFC insulin in front of the cingulate. But it is this question of, it would be really interesting, right, to do like sadness, take normal healthy controls, induce sadness in them, and see if you can flip it transiently to your question or not, right? Can we get a normal healthy control to do this? As you know, the early subgenual work, Mark George did this, and Helen, all these people did this. They do these, bold studies with sadness induction, and they were able to get, greater subgenual reactivity even in a normal healthy control. The hope is that it may not be actually an inducible phenomenon, but that's a great kind of question to kind of go forward with and try to sort out. Your other question, I've been thinking about a lot, and I've talked to Karen Parker, who's an autism researcher at Stanford, about this, but trying to figure out, is there a model in macaques or something like that where you could get in with a TMS coil like really young and try to do something there, and to try to change disease progression. And they have these sporadic autistic macaques, right? And can you get in like right at the beginning, right when they can kind of identify them and see if you can do something while the brain's very plastic, as a way of kind of justifying doing what could be a very intensive stimulation protocol to a 3-year-old or whatever, you know what I mean? And so these are kind of the ethical issues that you run into. But, if that vision that you're talking about is the vision, right? It's my vision too, right? And it's this vision of if you've got something safe enough that really is, you know, pretty side effect free. I mean, folks tell me they either are better or they're not better. They don't feel weird or any of that stuff. If you could do that, then you could have this sort of early intervention move to try to change life trajectory, because it is, you know, we're getting to these problems when they're already big problems. We're not really doing preventative medicine and psychiatry yet. And it's because we don't have the tools, to your point, to do that. And if we could, I think it's a beautiful story. It's just it's a very hairy one if you do it wrong. So, you know, people are always asking me if we're going to treat adolescents, and that's next. But it's like you got to kind of like, you have to kind of step it down, to your point. And I think that's definitely on that trajectory for sure.
Yeah.
Was there something online? Okay.
Sure. Thank you. As a student, trainee, researcher, and clinician, I really appreciated how the beginning of your talk talked about how all psych diagnoses are perturbations in distributed neural networks. So my question is, given that every diagnosis essentially implicates these huge networks, my understanding of the TMS literature is it mostly focuses on MDD and beginning to focus on OCD. And I'm curious if those are viewed as more biological, which doesn't quite reconcile with my understanding of biopsychosocial models of everything, or why these disorders, if there's the same targets in OCD, if there might be a future to expand TMS to other diagnoses, if they are all disorders of network-wide perturbations.
Yeah, no, it's a great question. I think, you know, so there's been a ton of work basically in, you know, in as a generality in kind of all psychiatric illness, right? People try to do TMS for everything pretty much, right? You know, I've heard people trying to, and I don't want to do these studies, trying to take, you know, antisocials in prison and try to treat them. And I mean, people are really thinking broadly about this, right? And so it's this, you know, depression makes sense as a first step because ECT was there, right? And because ECT has its issues, right? Only about 1.5% of people that meet Medicare, Medicaid criteria to get ECT in the United States get it, right? So you got a funding agency, you know, Medicare gets another plus point. Funding agencies, we're only going to pay for it. Nobody will do it. It's not standard of care because it can't scale, you know? And so, you know, there was an effort to try to, you know, depression was an effort to try to kind of replace that at a level, right? And so there's more clinical, to your point, more clinical pragmatic items kind of dictating some of this. What I can tell you is in the schizophrenia literature, there's no one target that at least anybody's found yet that can handle all schizophrenia. And so then the technology ends up being limited, right? You can knock down auditory hallucinations by inhibiting the left temporal parietal junction, primary auditory cortex, but they still have the cognitive, still have the negative symptoms. They still have, you know, some of the paranoia and all of that stuff. You're really just hitting one piece of it, right? Versus stimulating in the prefrontal cortex, you can improve the negative symptoms of schizophrenia, but you don't do anything for the auditory hallucinations, paranoia, and all that stuff, right? So you end up, you know, it's cool as a way of dissecting it, but you can, with the current technology, it's hard to make a treatment out of that treats schizophrenia. And then you end up getting stuck with, unless it's somebody who has like medication resistant auditory hallucinations, which is a thing, then it's hard to kind of understand how you scale that for schizophrenia, and people still have to do antipsychotics. Now there's all sorts of tech being developed, multifocal coils, where you stimulate in multiple brain regions at the same time that we will handle schizophrenia. I'm not saying it's an unachievable thing, but I would see myself as kind of a simple guy, and depression's a simpler problem than schizophrenia. Schizophrenia is a very hard problem, you know, but both network problems, right? It's less about that, and it's more about how do you take a bite of the apple as a scientist and really be able to wrap your arms around it with the technology that you have. And depression, at least for this subgroup of people, looks like you can handle it with a certain set of TMS approaches, you know, ours being one of them, maybe others, right? But yeah, that's really more of the goal. It's really just this goal of like, with the technology that we have and the more limited symptomatic scope of depression, going after something that you kind of wrap your arms around as a scientist. Does that make sense? Yeah.
And Nolan, here's a question from a clinician in our virtual audience about patients that she's treated who she observes to get much better, but they only acknowledge moderate improvement, if at all. Wanted to know what your experience was with that.
Yeah, it's a great thing, right? We see this with ECT. We see this with, for some people, middle of the week, early week saint, right? And I think it's a process that people go through. And, I'm sure you guys have seen this, right? Where the last person to know that the patient's not depressed anymore is the patient, right? The family's like, oh my God, you're like eating again, and you're going to the park with me, and you're smiling, and you're waking up early and all that. And then they're like, well, I'm still sad. And they're like, I'm the same. Right. And so we see that, and then people end up transitioning through that, and they get to this point where they're well. And I think that what you're seeing there is what I talked about earlier, Ned, which is this, if you look here, right? So that patient's kind of like in here or something, and then this is when they really know. And so from our perspective as a dosing thing, you got to kind of like go through that phase of like, everybody thinks you're better, but you're still like not sure, you don't agree or whatever. And then you get into the phase of you're feeling better, and it's really a dose phenomenon. We had some people, and this is really interesting, and people have run our protocol, have seen the same thing. So it's not just us, where people actually have this kind of, and this will be interesting, it's kind of emotional cathartic thing that happens. And it's like every time we see this, they remit the next day. It's really wild, actually, but they get inconsolable, and they're crying in the hallway and stuff. And and at first when we saw this, we're like, we're gonna have to admit this kind of freak, just keep treating them or whatever, And what we've seen is, and every person that's had that I've talked to, and you see this with ECT too, they'll say, I was numb and I wasn't feeling anything. And then I felt everything all at once, and then I felt better, And so it seems to me like the meta of that question is people kind of go through these stages. Some of them go through stages where they look great and nobody else, they don't think they're feeling any better, but everybody else thinks they look great. And then some people who have this cathartic thing, and then they feel better. They have to transit through all that stuff to really get back to a normal place. And because I think it happens with ECT too, it seems to not be a TMS phenomenon or a saint phenomenon, but rather like a trajectory of remission phenomenon.
And we see it with antidepressants as well.
Yeah.
With patients. Other questions?
Is it working? Okay.
Yeah.
Well, thanks again for the talk. Two small questions, maybe from the clinical perspective again. First, maybe I missed that, but the patient has also comorbidity with other diagnoses. How complex was their clinical presentation aside from severe depression and treatment resistant? And the other question is more about, so you talked about the waves of treatment and point 1, point 2, point 3. I was wondering if there's any, kind of convergent validity, if the other treatment of CBT or medication works through the same mechanism, and if you see changes in the DLPFC, ACC, pre-post other treatments, and broadly, if you think that there are like subgroups that in depression with different mechanisms.
Yeah, no, that's great. So one of my former colleagues at Stanford, who now runs a company, Alto Neuroscience, Amit Etkin's done a lot of that psychotherapy biomarker work. It's tough, right? Because what psychiatry is looking for is a treatment responsive, modality independent biomarker for psychiatric illness, right? Which is kind of your point, right? We need something that is independent of what the modality is and that responds to the treatment. We don't have it yet, right? And so some of the psychotherapy studies that have been done, they implicate structures like the left dorsolateral prefrontal cortex. Connor listened to this great trial as a MD PhD student where he, induced a kind of a state of helplessness in folks. And then you could see that DLPFC came offline in those patients, and so this idea that maybe CBTs bring people back online, so there are these ideas, but to say there's a kind of a validated, treatment responsive modality independent biomarker, we're just, to Ned's point, we're just not there yet, right? And we've got a lot of work to do. And a big part of it is this chicken egg thing, right? We kind of get into this thing where, like, in order to really get enough information at scale to do that, you need enough MRIs, and so it's like, and it's hard to get that funded. And so we end up, I think, having a hard time with that. And then we're every year figuring out new ways to analyze the same kinds of data. So my hope is at some point in the next, next couple of years, once we have this sort of data at scale, what Amit's doing, what various other companies are doing to try to get a lot of biomarker data at scale that could get FDA approval, then you'd be able to pick that out. But yeah, right now, I think we're kind of limited by the funding environment,
So I think we've reached the end.
I spill a little coffee. I don't know if somebody wants to.
Thank
somebody
you
has a
so much,
napkin at all.
Nolan. That was just wonderful. So we're gonna now have a period where there are breakout discussion groups, and they're listed here. So we're gonna meet for a little under an hour. Please be back promptly at 11:15. Molly Crockett's talk will be at 11:15. And go to whichever discussion groups you wish to go to, and you can start with one and transition to another during this period. So this is an opportunity to meet the speakers and talk to them in more depth. So please enjoy this, and see you at 11:15.
Hey, thanks, man. Yeah, thanks.